Hi, I'm Mia Arkles, and this is Colin Ordaway, and we do research for Dr. Amanda Harwood here at Alma College. So today we're going to be talking to you about our research we're doing, which is evaluating the toxicity of activated carbon to Eisenia fetida. So to begin, we're going to talk about what is activated carbon. So activated carbon is a form of organic carbon that is highly porous, which means there's a larger surface area because you have all those little pores within the particles. So with the larger surface area, you now have more active sites in which organic chemicals can bind to the carbon. Now, organic chemicals will bind to the carbon because carbon is very nonpolar. So now you have these organic chemicals that can absorb and bind to these active sites. Organic carbon is really common. So if you have like a Brita filter at home, the filter that's used to filter the water most likely has activated carbon in it. So let's kind of talk about how activated carbon works. So as we discussed, it attracts and holds on to organic chemicals. So because it is this nonpolar hydrophobic, these pollutants will bind to the carbon, and the carbon really doesn't want to let go because it's very nonpolar. So then you now reduce the bioavailability of these organic chemicals within the environment. Bioavailability is the amount of chemicals able to pass through a membrane or be absorbed by an organism. So now with the chemicals being bound to the carbon, they're no longer able to kind of interact with the environment. Now it doesn't get rid of the pollutants. It only sequesters it, but it's still not able to cause all these effects. So activated carbon is used a lot in soil and sediment remediation. What we mean by this is if you have an area that's contaminated with organic pollutants like pesticides, you spread this organic carbon, or this activated carbon rather, and it's able to bind to these chemicals, and now they're not able to interact with the environment. So in St. Louis, Michigan, along the Pine River, there is a Superfund site where the former Vesicle Chemical Corporation was. They were a large producer of pesticides, specifically DDT. So this site is contaminated, and downstream and within the floodplains are also contaminated. So currently there's a pilot study being done by the EPA, Dr. Harwood, and several others, where they're taking this 45,000 square foot test area, and they're adding activated carbon to see if it can be used for soil remediation for DDX. DDX is your DDT, and then when it breaks down into metabolites. So we're talking about this whole group of DDT and its metabolites when we talk about DDX. So what they're doing is they're adding the activated carbon to the sample test site, and they're monitoring the bioavailability of the DDX. So they tested before they added the carbon, 3 months and 9 months afterwards to see if the bioavailability reduces. And what they have found so far is that yes, the activated carbon does reduce the bioavailability of DDX in this area, which is great. But the community of St. Louis has expressed genuine concern that is the activated carbon you're using for the remediation, is that itself toxic? So that is what our research is about. It's about looking at the potential toxicity of the carbon, which brings us to our big research question. Is the activated carbon we add toxic to E. fetida, which are just earthworms? Now we're looking at earthworms specifically in this study because, well, to begin, earthworms are a standardized species. You have a standard set of methods established to test with these species. So if someone else is doing the research in a different lab, it's all standardized. So we're looking at the toxicity effects on several biological factors. Toxicity, something being toxic does not always just mean it's going to kill you. There's a lot of sublethal points that we can look at to see if it has these effects. So we do look at survival. Do the things die when exposed? But we're also looking at growth and reproduction within these species when they are exposed to different levels of activated carbon.
So the methods that we looked at is that we run a 28-day test. And so with the jars, we had 675 grams of wet reference soil. So what we did was we took commercial dry soil and added moderately hard water in order to simulate an experience of what it's like out in the real world. And for each jar, we had 10 animals. And so for our adult tests, we had 400 to 600 milligram adults that had a cotellum. The cotellum is the reproductive organ in earthworms. And with our juvenile tests, we weighed 200 milligram juveniles. And with these jars, we put them in an environmental chamber to have an 18-to-6 light-dark cycle at 20 degrees Celsius. And on top of our soil, we applied the exact same activated carbon that they applied at the Superfund site, aquacarb. So this was added topically in proportions to wet weight. So we added them respectively to 0.5%, 1%, and 2% of the wet weight. And daily we would monitor the moisture levels in order to keep it the same as it was day one. And when we look at our endpoints, we looked at a couple different things. We looked at survival. Did these worms survive over the 28-day period? We looked at growth. When we put them in the jars day one and we took them out day 28, did their weight change? Did they gain weight? Did they lose weight? And then we looked at their gut mass. So at the end of the test, we have a depuration period of 24 hours to make sure the worms empty their guts. We observe this to make sure that they are indeed eating and that they had ate. And then we administer reproduction. So in our adult tests, we would observe the soil to see if there were egg cases to make sure that they had been reproducing and that they were laying eggs. And with our juvenile tests, we would look for the development of the cotellum as seen right here.
So as we begin to kind of look at our results, our preliminary findings, we have begun to find that the mortality rate is really low. There's not really an effect of this carbon, and it's not really killing the worms. We also have observed that there is no significant weight loss between the concentrations and the controls after the test is done, as well as no reproduction inhibition. If you look at the image here, you can actually see the egg casings of the worms after they had reproduced. And as we were breaking down the test, they begin to hatch on the table. So you see all these little baby worms coming out of the eggs, which is a really clear sign that there is no reproduction inhibition at all. And this was in our higher concentrations as well. So that's a great thing to find. So the concentrations we had been using were to simulate what would be experienced in the pilot study. These lower concentrations, you're just kind of spreading it across a field. Based on our preliminary findings, we wanted to see, okay, is there a concentration in which we will start to see effects? So currently, we are in the middle of doing tests with higher concentrations. It's the same methods, the 28-day tests with adults, using the same granular activated carbon, except this time we're homogenizing the mixture instead of just adding it as a topical layer, just to kind of see if it makes any difference. And we are now looking at these higher concentrations of 5, 7, and 10% by wet weight. And as we just now begin to break down these tests, so far there is no mortality in any of the concentrations, which is leading us to kind of conclude that our hypothesis was initially correct, that activated carbon is not toxic to these earthworms, but we are still continuing this research. And then this kind of just leads into our future work that we could potentially develop, which we could look at different types of carbon. So by this we mean the particle size, it could be different brand, washed versus unwashed carbon, which has to do with acid washing for effectiveness. We could look at even more concentrations. We could begin to look at egg hatching and survival. So by this, it's basically like a life cycle test where you have the worm that's exposed to the carbon, they lay eggs there. Okay, now are these eggs gonna actually survive into viable worms, or are they not gonna survive at all? That would be very interesting to look at. We are currently looking at topical versus homogenized applications to kind of see if there's any difference. And finally, looking at other species would be quite interesting for future work. Earthworms are invasive species, but they are also beneficial. And there might be more ecologically relevant species to look at for this study that may give better information. This could be other invertebrates. We could begin to look at aquatic life in ecosystems, or even plants. So that is kind of what we've been doing, kind of what we plan to do in the future. Thank you so much for listening, and if you have any questions, always feel free to ask. Thank you.